Welcome to the next video in our lesson on the autonomic nervous system, where we're going to continue this survey of the organ systems and the effect of the autonomic nervous system upon them and the mechanisms behind them, okay? And again, we're trying to establish some general principles that you can maybe apply to other tissues. We did the heart. In the last video, we have the muscarinic and beta receptors there. We don't have the alpha. At least it's not significant for our purposes if their effects aren't. In this video, we are going to cover arterioles of skeletal muscle. Okay, generally speaking, you can apply what I'm about to tell you to all the arterioles throughout the body in a general way. There's variations between regions, but more or less mechanistically they act the same. Okay. Arterioles have all four of these receptors. They have alpha 2, alpha 1, beta, and muscarinic. The muscarinic causes dilation, the beta causes dilation, the alpha 1 and alpha 2 cause constriction. So we're going to talk a little bit about how all these receptors operate, and then at the end I'm going to talk to you about how the system operates with all these receptors acting together. Okay. But first, let's cover the individual receptors. So first of all, let's cover the alpha 1 receptor. Okay. Odd-numbered alpha receptors and odd-numbered muscarinics all respond in the same way in tissues. That is, I should say, they all use the same second messenger system. It is the IP3 system. Okay, so in this case, for the alpha 1, we have alpha 1 binding norepinephrine. That activates a different G protein from the ones we've discussed so far, GQ. GQ stimulates phospholipase C. Okay? Phospholipase C causes the generation of IP3, inositol triphosphate. Okay? And I will always refer to these by their acronyms. All right. The IP3 binds to a channel in the SR membrane. It's labeled ER here for our purposes. They're the same thing in smooth muscle. ER, endoplasmic reticulum, SR, sarcoplasmic reticulum. Sarcoplasmic reticulum is really just more developed endoplasmic reticulum. So in this sort of a cell, the names are interchangeable basically. IP3 binds to this channel called the IP3 receptor, and that channel opens and causes calcium release from the SR into the cytosol. From there, we're going to proceed through EC coupling in smooth muscle in the same way that I described it in the smooth muscle lesson. Let's review it. Whoops, calcium goes up and binds to calmodulin. So, calcium calmodulin activates myosin light chain kinase. Myosin light chain kinase phosphorylates myosin light chain, that is myosin in smooth muscle, and that causes contraction. So your alpha 1 receptor causes contraction through this mechanism. And I'll tell you, generally speaking, when we talk about alpha 1 receptors, everything up into calcium is going to be the same. And the same thing will be true of odd-numbered muscarinics. The pathway is basically the same, everything up through calcium pretty much the same. Okay? All right. So that is a general paradigm, and we're going to revisit it. Now, we also have beta 2 receptors in vascular smooth muscle. Now, I will tell you, all beta receptors have the same second messenger system. So in the previous video, we went through beta 1. What was the mechanism for beta 1? What did you get? You got stimulation of GS, That stimulated adenylyl cyclase. That increased cyclic AMP. That activated pKa. That caused pKa to phosphorylate various targets in the cell. And just like the SA node and the cardiac muscle was basically the same second messenger system, activating pKa with different targets, the same thing is true here. In this case, our target is going to be myosin light chain kinase, not the L-type calcium channel and phospholamban and troponin I and things like that. It's a different target, but up to pKa, it's the same second messenger system. All you have to really know is what the targets are. And in this case, the target really intimately associated with smooth muscle contraction, which means that it kind of makes sense that that would be the target for it. So let's go through it. We have norepinephrine binding to beta-2 receptors. Beta-2 cause increased cyclic AMP. Increased cyclic AMP activates pKa. pKa will phosphorylate myosin light chain kinase. And when it does that, it inhibits myosin light chain kinase. That's what this minus sign means. It makes it less active. Less active myosin light chain kinase means less phosphorylated myosin light chain, and that means relaxation of vascular smooth muscle. Beta-2 receptors cause relaxation of vascular smooth muscle, okay? All right, so having gone through that, what about the alpha-2s? Well, I will tell you, even-numbered alphas and even-numbered muscarinics have the same second messenger system. So what was the second messenger system for the M2 receptor in the heart? Because this is the alpha-2, they're both even-numbered. Well, we activated GI, inhibitory G protein. That G protein caused inhibition of adenylyl cyclase, that caused a decrease in cyclic AMP, that caused a decrease in pKa activity. So what do you think the alpha-2 receptor is going to do to the response here? Correct. I hope it's correct. Okay, it's going to decrease the cyclic AMP, decrease the pKa, and it's going to increase myosin light chain kinase activity. Because increased pKa activity caused phosphorylation of myosin light chain, and that caused relaxation. Decreased pKa will cause myosin light chain kinase to be less phosphorylated, and it will therefore be more active. More active myosin light chain kinase gives you more phosphorylated myosin light chain, and that gives you an increase in contraction of your vascular smooth muscle, okay? Should make sense, and these do the opposite things, and they do, okay? And you should note, same second messenger system as we had in the heart tissue. It's just that the target for pKa is different, and it's a target that should make some sense, okay? All right, what about the M3 receptor? Odd-numbered alphas and odd-numbered muscarinics have the same second messenger system. We just did an odd-numbered alpha. Okay, it acted through IP3 to increase calcium by releasing calcium to the ER. Now, what's going to happen with M3 is the same thing, only the receptor is located in the endothelial cells, which line your vasculature. So this receptor is not in the smooth muscle membrane, it's in the endothelial cell membrane. But from here, all the way to here, it's the same thing. Activation of GQ, increased PLC, increased IP3, and increased release from the ER, this time in endothelial cells, which increases calcium. And up until here, it's exactly the same. Now, the target in endothelial cells is a different target. It's actually nitric oxide synthase. Nitric oxide synthase, actually, it's calcium binding to calmodulin. which activates this. And in response to this, you increase nitric oxide, which is a gas, okay? NO, nitrogen and oxygen. The gas, as you know, most gases are permeant to membranes. Nitric oxide, no exception. It will go from the endothelial cell to the smooth muscle cell, across the membrane, and there it activates guanylyl cyclase. Adenylyl cyclase increases cyclic AMP. Guanylyl cyclase increases cyclic GMP, and it converts GTP to cyclic GMP, okay? Now, cyclic GMP does a number of different things that will ultimately end up relaxing vascular smooth muscle. First of all, you have an increase in myosin light chain phosphatase activity. Myosin light chain phosphatase, as you might guess, acts on phosphorylated myosin light chain, and it cuts that phosphate off and it generates myosin light chain. So what you find in smooth muscle is you've got some unphosphorylated myosin light chain and you've got some phosphorylated myosin light chain. And how much of each you have is dependent upon how active myosin light chain kinase is, because it phosphorylates MLC, and how active this phosphatase is because it's converting this back to unphosphorylated MLC. So increased cyclic GMP increases the phosphatase activity. That generates more of this, and that causes relaxation, okay? You have cyclic GMP, which activates protein kinase G, PKG, just like cyclic AMP activates protein kinase A. This activates protein kinase G. That phosphorylates phospholamban in smooth muscles, which increases calcium uptake and decreases cytosolic calcium. Decreased calcium binding to calmodulin, decreased calcium calmodulin, less active myosin light chain kinase. Less active myosin light chain kinase means you have more unphosphorylated myosin light chain and you have relaxation, okay? And finally, it increases the permeability of the membrane to potassium. So what it does is it opens a channel that lets potassium out. So you have positive charge going out of the cell and you cause the membrane voltage to go down and become more negative. That decreases the activity of voltage-gated calcium channels, which you will find in the membranes of these smooth muscle cells, and that will decrease cytosolic calcium. Decreased calcium calmodulin, decreased calcium calmodulin, decreased myosin light chain kinase activity, decreased myosin light chain kinase, more myosin light chain, less phosphorylated myosin light chain, and relaxation, okay? All right, you may wonder why I'm torturing you with these. These are drug targets. The pharmacologists are going to take you through this, and they're going to take you through some drugs, and you're going to find drugs that target this pathway. You know, basically, you're going to find out there's a pharmacological correlate to all these. If you look deep enough, you'll find it, and I know they're going to teach you some of them, because I've seen the notes, okay? All right. So this isn't, I know it seems like a waste of time, it's really not. At bare minimum, you should be getting the idea that these second messenger systems are predictable, and that means the responses in the tissues will be predictable, okay? All right, so you might ask yourself, I would ask myself if I were you, what is going on here? Because if I stimulate the sympathetic nervous system, and the only input to vascular smooth muscle is sympathetic, I should have sort of pointed that out. This ACH that binds to this M3 receptor on endothelial cells, it's coming from sympathetic nerve endings. The innervation to vascular smooth muscle, most places in the body, almost everywhere, with rare exceptions, the innervation is sympathetic. This ACH is being released from sympathetic nerve endings. This causes sympathetic cholinergic activation, okay? All right, so having said that, you've got the sympathetic nervous system, and you've got 4 different receptors, all of them binding neurotransmitters, and they're all doing different things, right? You have sympathetic nervous system stimulation, It's causing the alpha receptors to cause constriction. At the same time, it's causing the beta receptors to cause dilation. It's causing, your muscarinic receptors to to cause a dilation. So what's going on? We have competing effects here from the same autonomic system. So how does this all work? Well, that is described in this graph. I have 4 panels here. Each of these represents flow on the y-axis over time. And what these are vessels that an experimental biologist isolated with the nerve. And so what they're doing is they're stimulating a sympathetic nerve under different conditions. So this is your control condition. This is just the section of vasculature with the nerve intact, and what you're doing is you've got flow through that vasculature, and you're measuring the rate of flow. So you stimulate the sympathetic nerve, and what happens is flow goes down. So that should automatically tell you that the dominant steady state sympathetic response is vasoconstriction. And that is what your autonomic nervous system does to arteriolar smooth muscle. All right? So that is number 1. You have 4 different receptors. They're all doing the same thing. The dominant response is constriction. Now, and we know it's constriction because the flow is going down, which means we're increasing the resistance to flow. Okay? All right. So, and we know that this is due to the alpha receptors because of the 4 receptors that we looked at, they were the 2 that caused restriction. Alpha 1 and constriction, alpha 1 and alpha 2, all right? So let's block those alpha receptors and see what happens. Now we stimulate our sympathetic nerve, and we have an alpha blocker on board, which is blocking the alpha 1 and the alpha 2, and now look, flow goes up. And we get an increase in flow, which means we have vasodilation now. So our alpha receptors were causing this constriction without them active because we blocked them. We get an increase in flow, vasodilation. Okay, what's the vasodilation due to? Well, we can block acetylcholine receptors, the M3 in the endothelial cells, and now we stimulate our nerve. We get a little bit of an increase in flow, but not as big as it was when we had our muscarinic intact. Okay, and then finally you block the alpha, the muscarinic, and the M3 and the beta receptors, and now you stimulate your sympathetic nerve and everything goes away. Okay? So you might ask yourself, why are these things here if they're not really part of the overall response? Well, answer number 1 is that the muscarinic response, the M3 receptor response, kind of is a part of the response. And the reason is because it becomes relevant when you're exercising. If you exercise, what you get is you'll get vasoconstriction in your body because the sympathetic tone will go up. You'll get stressed. I get stressed just thinking about it. Okay, exercise not my thing. So the net effect is going to be vasoconstriction to that response. Okay. But before you get this vasoconstriction, what happens in your arterioles, particularly those in skeletal muscles, you get a little bit of a dilation. It's called the anticipatory response. And it's like somewhere in your brain it knows, okay, he's getting ready to exercise. We got to get ready for, heaven help us, we got to get ready to do this. So you get a little spurt of extra blood into your muscles and into your tissues in anticipation of the exercise. And so you get an anticipatory response, which is a small transient dilation. And so you get a dilatory response right before you start to exercise. And then after that, the alpha receptors kick in with just a very slight delay, and the net effect is vasoconstriction. So the net effect of sympathetic nervous system on arteriolar smooth muscle, vasoconstriction. You get a transient increase in flow that precedes that that is dependent upon your M3 receptors, the muscarinic receptors, okay? So they actually have kind of a role in terms of dilation. The other thing is, why the beta receptors are there, who knows? I don't know. I don't think, to my knowledge, nobody knows why they're there. But it is fortunate for us that they are there, because you can take advantage of these pharmacologically, and you can add a beta-2 agonist and get vasodilation. And so these are receptors that can be targeted. We don't know the physiological function, but we know they can be targeted. All these can be targeted with drugs, okay? All right. The other thing about exercise I want to bring up, and this may have occurred to you already, I said you're exercising, your sympathetic activity is high, you get vasoconstriction. And you're thinking, why in the world would I want to get vasoconstriction to my muscle when I'm exercising? And the answer is you don't. So here's what happens. This little purple thing is exercising. And it's exercising and it's curling, it's lifting weight with its left arm. And you can't see its right arm, but it's hanging by its side and it is not doing anything. Okay? It is worthless and weak. So right now I'm exercising. And I need to get extra blood to my exercising muscle because my left arm is eating up ATP and it's eating up oxygen and it needs that extra blood. It deserves that extra blood. It's working for it. My right arm, as you look at me here, is worthless and weak. It is sitting there by my side not doing anything. It does not deserve extra blood. Why should I dilate the vessels going to the muscle in my right arm when it's not doing anything? That is a waste of blood. Okay? So what's happening in my body, and my left arm is actually getting tired now because at my age, this is what passes for exercise. What's happening is metabolites are building up in the muscle in my left arm. And as those metabolites build up, they cause vasodilation. They cause a local dilation. The local response, the local metabolic response, the effect of local metabolites upon arterial or smooth muscle dominates over the extrinsic autonomic nervous system response that usually causes vasoconstriction. So the sympathetic nerves in this arm are actually being stimulated. They are actually trying to cause a vasoconstriction, but the local metabolites that are building in this muscle are overriding that response and causing dilation and causing this arm to get more blood. On my right side, vasoconstriction is taking place because no metabolites are building, and that's a good thing. Why should I shunt blood to my right arm when it's not doing anything? It's not earning that blood. My left arm is. My right arm doesn't need it, so I can constrict to the blood supply in my right arm, in my skeletal muscle, and cause a change in total peripheral resistance that way and try to keep my mean arterial pressure up by constricting here. But I don't try to keep my mean arterial pressure up by constricting here. That would defeat the purpose. The purpose is to get more blood flow to the exercising muscle, not to the resting muscle. Okay? So when you exercise, the local factors, the local metabolic factors dominate over the sympathetic nervous system. So the muscles that need blood get it, but only the muscles that need blood get it. Okay? All right. That's all I've got for the vascular smooth muscle. I know that was a lot, and I think it's all downhill from here. In the next video, we'll do GI and we'll do the exocrine glands, the sweat glands, the salivary glands, and we'll finish this lesson up. Thank you for your attention.